And I'll be deferring to, we've just gotten a notice that our meeting is being recorded, just as a reminder for everybody that we will make this available once we've tidied it up, and then we will be posting it, and then we'll send out a notice to all of our board members and our attendees. I think. All right, very good.
All right, thank you, Gigi. I'm going to set just a couple of ground rules for everybody, and not even ground rules. It's really an invitation to engage with. I'm sorry, I work with a bunch of physicians, I have to say Dr. Marks with Peter. But we will, Peter has prepared a series of slides for us in the beginning. And I think Joe is going to do a formal introduction. If we were all together in person, we would walk around the room and let you all know who we are and what our organizations are. That's very hard to do in the Zoom environment. So I think what I would ask is if someone asks a question later, that you would identify your organization so that our speaker can get a sense for who's in the room. You do have representatives of a variety of the companies and industries in Montgomery County, not just healthcare, but also scientific enterprises and hospitality and aerospace or defense programs, human resources, so the whole range of things. What I wanted to let everybody know is that Joe and I together are going to be monitoring the chat. So if questions come up, while Peter is speaking, if you want to pop those into the chat or later on as we're moving through, and then Joe and I will try and make sure that those get answered for everyone. So thank you, Joe.
Yes, thank you and welcome everybody. So we are privileged to have with us today as our guest speaker, Dr. Peter Marks, or Peter as he prefers. He's the director of the Center for Biologics Evaluation and Research, or CBER, at the FDA. The center itself is responsible for assuring the safety and effectiveness of biological products, including vaccines, a timely topic for us today. They also cover allergenic products, blood products, cell, tissue, and gene therapies. Dr. Marks and his staff are committed to facilitating the development of biological products and providing oversight throughout the product lifecycle. Dr. Marks, or Peter, welcome. If you could please do us the favor of introducing yourself much better than I just did, and share with us a little bit more about your background and career trajectory into the FDA and CBER.
Okay, so thanks very much. I'll fill in the little bit of the blanks. So I'm a New Yorker by birth. I was born in Brooklyn. I grew up in New York. Was afraid to leave Manhattan Island till I was about close to 30 because I went to Columbia and then to NYU for medical school and graduate school. And I was finally convinced to leave the island of Manhattan to go to Boston for residency and fellowship, and lived in Boston for about 12 years. Before I worked then took a job in the pharmaceutical industry, worked 2 different jobs in the pharmaceutical industry. First at a large biotechnology company, Genzyme, and then at Novartis, a large pharmaceutical company. Prior to returning to clinical medicine about 15 years ago, at which point I was at Yale and ran the leukemia service and then ultimately the clinical service on the cancer hospital. And then about 10 years ago, close to it now, I was recruited to be the deputy center director at the Center for Biologics, which let me actually run our research labs and help run our research labs and do some really neat regulatory work, and have been doing that since. Became center director about 5 years ago, and for the past year and a bit have been living the dream of COVID-19 in most of the time either at FDA or in my basement here. So I look forward to telling you some of the non-COVID-19 stuff we do at some point, but I think today I was going to tell you about what we've done to help facilitate the development of COVID-19 vaccines. And I put together about a 15-minute slide deck to try to answer some of the more common questions. You might know all the answers, but I'll try to tell it to you and then be happy to take as many questions and answers as you have. Does that sound good?
That sounds fantastic. So I believe that we're going to be running the slides for you.
Okay.
They'll come up and
Sounds
project
good.
on the screen, and you can take it from there.
Great, thanks. Okay. So if I can have the next slide. Um, you know, vaccine development is something that in non-pandemic times is somewhat of a backwater. Companies don't really get all that excited about developing vaccines in general because they are not big money makers. You need to have a large infrastructure to make them, and yet much of the time that infrastructure might kind of sit fallow, as it does sometimes for influenza vaccine or certain other vaccines. And so it's a very de-risked process when you're developing a vaccine. So it goes in very orderly stages, and one usually does not start to really scale up the manufacturing of a vaccine until very late in the game. And that means that sometimes the launch of these vaccines can be delayed, even just because it takes time to scale up and get the vaccine ready to go. Now, in the case of a pandemic, proceeding in very orderly, measured stages where after you get one result, you think about it, and then you go to the next result. Let's try to get the next result. That's maybe not the best way to get an answer fast. And so if I have the next slide, what was done for the COVID-19 vaccines was to build upon the scientific knowledge that we had to get vaccine candidates as quickly as we could. And that was those who may have seen the pieces done on Moderna and Pfizer. Those people worked very quickly to get candidates and then get them into clinical trials that had multiple phases condensed into one. So phase 1, 2, or phase 1, 2, 3 trials. And that's actually the Pfizer trial, the large trial that led to its emergency use authorization was a phase 1, 2, 3 trial. So one doesn't have the stopping and starting. And then the other piece that was done was rather than waiting to start up manufacturing was scaled up from, was from the start, knowing that, well, the worst thing that would happen would be at the end of the day, we would have a process that, and probably some vaccine that we wouldn't be able to use. But the bigger danger was having population at risk without a vaccine. And so this whole concept of manufacturing and risk and proceeding rapidly with clinical trials, that was what was put into, at the time, Operation Warp Speed, and kind of moved forward. And it did accelerate vaccine development because it took less than a year from the vaccine candidate to actually have vaccine being distributed. If I go to the next slide. So what is getting distributed? Well, what's getting distributed right now is under emergency use authorization because there was not time to get it out there under a biologics license application. Most vaccines, and the vaccines that you receive all the time from the doctor's office, have been licensed by the FDA, which means the manufacturers have submitted a biologics license application. We use the Public Health Service Act to issue licenses that say the vaccines are safe, pure, and potent. They do what they're supposed to do. And in order to get there, one has to have evidence from adequate and well-controlled clinical trials. I have the next slide. The emergency use authorization authority that FDA has was put in place so that we would be able to address disasters, natural or man-made, that might occur. And it allows us to address chemical, biological, or radionuclear events. And in doing so, once the Secretary of HHS has declared a disaster, we can make products that might otherwise have been investigational available for use if they may be effective, if there's not an alternative that's available, and if the known and potential benefits outweigh the known and potential risks. Now, for many people who start out being a little vaccine hesitant to begin with, maybe effective sounds a little bit weak. And we understood that. And so if I have the next slide, when we went to start to study these vaccines, knowing that there would be a lot of people to potentially study, knowing that there was a fair amount of COVID-19 around, we felt that large clinical trials could be conducted. And so we put in our guidance from FDA that if we were going to authorize one of these vaccines, it would have to be on the basis of a clear and compelling demonstration of efficacy from a large, well-designed phase 3 clinical trial. that's very similar to what we require for a biologics license application. The one difference is that although you can get efficacy quickly when you have enough cases, the duration of safety follow-up is something that one needs time to get. And because of that, we put a minimum time which we needed to follow people, which was a minimum of 2 months, meaning of 2 months follow-up to deploy these under EUA. But we would make up for this by using enhanced post-deployment surveillance. And then the totality of this would be done transparently, where you probably are aware we took these vaccines that we've authorized to public advisory committee meetings. The documents that the companies submitted or parts of the documents are posted on the web. Our decision memos are also posted on the web. So we've tried to be as transparent as we could be. Have the next slide. And so what have we ended up with? Right now, there are 6 candidates that were either sponsored by the government or for which the government has bought large quantities of vaccine. Those include the 2 mRNA vaccines, the Pfizer, BioNTech, and Moderna mRNA vaccines, 2 non-replicating viral vector vaccines, one from Janssen. And I should say that the 2 mRNA vaccines, as you're all probably well aware, are 2-dose vaccines given either 3 or 4 weeks apart. The Janssen vaccine is a single-dose vaccine. There is another adenoviral vectored vaccine from AstraZeneca, and that is still awaiting regulatory submission here in the US, whether it will be an emergency use authorization or a biologics license application. And there are 2 other vaccines that are in development that are winding their way towards an emergency use authorization or biologics license application, whichever they decide to submit. One from Novavax and one from Sanofi. Those are the more traditional protein subunit vaccines where you just give the protein that you want to make an antibody to along with an adjuvant. It turns out those happen to be the slowest because they took the longest to get the manufacturing and other issues done, but they're all potentially very useful if they're shown to be efficacious and safe. So the next slide. So let me tell you about the three authorized vaccines. So the 2-dose mRNA vaccines were studied along with the Janssen vaccine. They were studied in different trials, but all of those trials enrolled somewhere between 30,000 and about 43,000+ people. They were 1-to-1 randomizations, which means half of the people got the vaccine and half of them got a saline placebo. The demographics of these trials mirrored the demographics of the United States, and they included underserved minorities as well as Native peoples. There were American Indians and Native Alaskan peoples enrolled in them. And about 20 to 25% of the individuals were over age 65, because we knew that that really COVID-19 had a really disproportionate effect, particularly in people over 75, but also people over 65. And the next slide. The vaccines were all highly efficacious. And in fact, the top line message here is that all 3 vaccines prevent death and hospitalization from COVID-19 equally well. It is true that the Pfizer, BioNTech, and Moderna vaccines do a bit better against milder forms of COVID-19 than the Janssen vaccine, but there's a trade-off there of 1-dose versus 2-dose, and the side effects profile of the Pfizer, BioNTech, and Moderna vaccine gives a little bit more fatigue, as I'll show you in a moment. If we can have the next slide. If one looks, the most common side effects, these are remarkably safe vaccines. The most common side effects are short-lived, and although many people don't like them because they're a pain in the site where you get the vaccine, or kind of feeling flu-like or fatigued for a day or 2 after, those are really almost always self-limited side effects. They're particularly more present after the 2nd dose of the vaccines, and they're less common the older you are. And whether that's because of the extent of the immune response or the extent of the off-target immune response. We just don't know at this point. If I have the next slide. What I was saying was that this study was done in a range of children that was reflective of the population. It was a diverse population, perhaps not quite as diverse as the adults, but reasonably so. If you have the next slide, please. Okay, so what was really impressive was the immune response in 12 to 15-year-olds was as good or actually better than 16 to 25-year-olds. Some of that might have to have done with the fact that the same dose of vaccine was used, and 12 to 15-year-olds are generally a bit smaller than 16 to 25-year-olds, although each of you may have your own experience with your own children and know that that is not invariably the case. But excellent immune response, if I have the next slide. And importantly, because this study was done when we had COVID-19 rampant, even with just 2,000 or 12 to 15-year-olds available for efficacy study, they compared 1,005 vaccinated 12 to 15-year-olds to 978 unvaccinated or saline vaccinated placebo 12 to 15-year-olds, and no one who got vaccinated had got COVID-19. 16 of the 978 that got the saline placebo got COVID-19. So an excellent rate of protection here, and virtually 100% in a small sample here. So a vaccine that seems to be working quite well. And luckily also, if I have the next slide, the safety profile of the vaccine is, if anything, a little better than in older individuals, 12 to 15-year-olds and 16 to 25-year-olds. You can see the percentages of these side effects are very similar. It seems that 12 to 15-year-olds are quite happy to be able to get vaccinated because they're excited to be able to potentially get back to the things that they want to be doing. The next slide, please. So basically, we now have the latest news, obviously, as of yesterday. You probably heard that Moderna, the Moderna study in 12 to 15-year-olds was also seemed to show that the vaccine appears to be safe and effective. We have not reviewed those data yet at FDA, and obviously they'll be submitting them. And when they do, hopefully we'll again see a second mRNA vaccine with that larger expansion to 12 and up. Now, throughout this, one of our major concerns, not just for children, but for everyone, is that we make sure that there aren't any untoward safety events that we can avoid or can warn people about. We have done this through a dual method here of looking at safety events that are reported to us through our vaccine adverse event monitoring systems, and also by using large databases to go looking for adverse events. The Sentinel system was put together now over a decade ago to look at adverse events in kind of a distributed database model using claims data. We've now enhanced that, and it's called BEST or Sentinel BEST, where we have a claims-based database that's also connected to the electronic health record for tens of millions of records. So we can look to see if certain signals are present or not. It's a little complicated with COVID-19 because we have to go get the states to give us the records of who's been vaccinated, because many people are getting vaccinated in mass vaccination campaigns, but it's worth the effort to make sure that we understand the safety profile of things. Have the next slide. These systems are working. They're working because they're picking up things such as the thrombosis thrombocytopenia syndrome, which was reported several weeks ago. It's what led to this pause in use of the Janssen vaccine and to some cautions to providers. These rare blood clots, these are things that you just don't see normally. Thankfully, we identified this. We've essentially warned the group that is most at risk, so they know what to watch out for and can make an informed decision about whether they want this particular vaccine. So it turns out that this particular side effect seems to be most common in women age 18 to 50 years. You might ask why. It's probably because this is an off-target immune response, something a little bit like an autoimmune response. And autoimmune responses are more common in women of childbearing years. Finally, there were, oh, by the way, it wasn't just women. There was one case seen in a man. The next slide, please. Finally, I just want to say that we obviously have to think ahead because it's wonderful that we now have more than 50% of adults in the United States are fully vaccinated with both vaccines. That's, or at least one dose of the Janssen vaccine. So that's great. But we still have a significant portion of the population that is not vaccinated, which means they are still susceptible, as probably some of us who have been vaccinated, to some of these variants, depending on how good an immune response we make. And so we need to keep track of the variants. And unfortunately, when you have viruses that's circulating in this kind of biomass, we have to deal with this. So we are keeping track of these in conjunction with our colleagues at CDC and also at the World Health Organization. And we're ready and prepared if we need to switch vaccines. The really lucky thing to date is that these mRNA vaccines seem to, and for that matter, the Janssen vaccine too, seem to have raised such good immune responses that the titers of antibodies that one gets to these vaccines after 2 doses of the mRNA vaccines are high enough that they seem to take care of all of the variants that we know to be circulating right now. Granted, they don't work quite as well in the laboratory, but for the standpoint of protecting you against death or hospitalization, they seem to do quite well. So if I have the next slide, we still yet, even with that said, we need to be prepared for what could happen because things could get out of control. So we have put out guidance. We've helped manufacturers understand how we would go about altering the vaccine if we need to address a changed variant that would lead us to need a new vaccine. Now, the good news is because they'll be working off of the same vaccine platforms, they won't have to do big clinical trials. It'll be small clinical trials, and some of them are already working on having kind of a stock of vaccine candidates that they've tested already, so that if we need to go with them as a booster, they'll be ready. So we need to try to still stay ahead of this virus because it keeps throwing us for a loop. And just when we think we're, you know, I was so, I'll tell you a little thing about myself. I was so uncoordinated as a child that it was considered like something you would go celebrate if I could make a basket on a basketball court. But I could run really well. I was good at running. And I was good at running because I understood one of the key things to running, which is that even if you're far out ahead, as you get to the finish line, you always try to accelerate as fast as you can because you don't want to find out and be surprised. And I think in this COVID-19 pandemic, we're a little bit in the same place. It's very easy to get complacent now that we have vaccines, we're taking off our masks. But as a public health professional, I am paid to worry for you and stay up at night worrying about what could happen here if we don't, you know, as we get closer to the finish line, because we'd really like to see this thing finished off. Can I have the next slide? So as we move forward, obviously it's a wonderful thing, and the story of the COVID-19 vaccine development and deployment is pretty impressive. But I leave us with the sobering thought that it isn't over until it's over, and some of us will continue to be worried. We get paid to be anxious. It's great about what's going on here. So I'll stop there. Thanks very much.
Thank you, Peter. You are not alone in your level of anxiety, because we had a number of people bring in some questions ahead of this, and they'd all sort of fall in that general category. I mean, it's an amazing scientific achievement to have had vaccines available already, you know, with roughly a year after we were all started growing to awareness to this. But as you said, everyone's looking to the future. And on that respect, I mean, what role does, we're going to talk about boosters, we're going to talk about, I guess it's BLA authorization versus EUA authorization. But before that, can you tell us a little bit, what role does FDA play in this ongoing debate about whether the best way to get in front, to outrun the pandemic, is through vaccination or in combination with achieving natural immunity? And just where is FDA in that?
Yeah, so FDA will, I think we would probably be one of the public health agencies, the voices at the table, arguing that probably the way to get there is not through natural immunity. And the reason for that is that many people will die along the way if we go there via natural immunity. And we've also learned something else, that if you develop natural immunity against the original Wuhan strain of COVID-19, you may not be protected against the variants that are coming from either South Africa or India at this point. And though maybe you'll be protected enough so that you won't succumb to COVID-19, bad COVID-19 is not something that anyone wants. It's a drag on the health system. And if you get bad COVID-19, you probably can also get long hauler syndrome. And that type of brain fog, for those of us who do executive jobs, that's certainly not something that we want, right?
Absolutely. So if we're looking at
no one doing any job wants that, right? But I just think of it through my lens, that if I've had people describe to me what they feel like, and I couldn't imagine doing, going through the day like that.
Absolutely. So if we're in this vaccine world, some of our companies have talked about whether or not to require, and even in the healthcare organization I work for, whether or not to require our employees to get vaccinated. And some of that hinges, the reluctance hinges on the fact that these vaccines are under EUA right now. So what do you think the future is for when we might see the Pfizer and Moderna and the Janssen vaccines receive their biologics license?
Well, so it's a matter of, I can say it because it's a matter of fact that Pfizer finished submitting, they announced that they finished submitting their BLA. So their BLA is now under review by the agency. These are big things to review. It's not like something we can turn around like an emergency use authorization. It's not, can't do it that fast. But I would not be surprised if sometime this fall we start to see the first biologics license applications approved. So I think this summer would be overly optimistic, but I think anytime this fall is probably probably realistic.
Mm-hmm.
And at that point, you're right. Things like the Department of Defense can require vaccination at that point, and I suspect more businesses will tend to think about this. This is obviously a very divisive. Incredibly divisive issue here. It's one of my reasons why when asked, should we have mandatory vaccination, my answer is always to punt and say, look, we should try to make a compelling enough case that everyone wants to get vaccinated, not that we have to force them to get vaccinated, because I think if you answer people's questions, and part of this is that there is still an amazing amount of denial that this is a terrible disease, because people who haven't seen it or haven't been touched by it don't realize quite what this is. But I think when you can try to make it real for people, they sometimes take it more seriously.
Yeah, I think some of the public service announcements that show people coming together and hugging again, and
Yes.
Grandparents and kids are very effective.
Yep.
So we're in this world then. What's the current thinking about booster vaccines? Is this looking more and more like it's going to be the 10-year tetanus booster or the annual flu shot variant? Do we know yet?
Well, we don't know for sure yet, but we have some hints. And I think the hints are, it may, between those, that's a great example you just gave, because it lets me be really fuzzy in the middle. It's probably closer to the, it's probably closer to the influenza booster than the tetanus, but I can't say how much closer. It may be a year, maybe 2 years. We just don't know exactly, but we're probably going to need boosters against these within somewhere between, I bet you, a year to 2. But again, I can't say for sure because we're still getting the data, but that's where it looks like it's headed.
Yeah, it certainly would be wonderful if we're not forever tied to the original vaccine. You know, I'm a Moderna girl and my husband's a Pfizer man, and you know, whether the booster has to come from the same company. I can imagine from the kind of public health campaigns we're going to want to do that it would be most useful if it's a single, you know, that there's some opportunities to mix and match.
The mix and match studies are getting done. So you're asking about mix and match, and those are getting done.
Great. Great. One more, you know, this was such an amazing scientific breakthrough, this mRNA technology that came into deployment with the Pfizer and Moderna vaccines. A little, as you said, wanting to talk about other things besides COVID. Are we going to see this? Can you tell us about some other candidates for where we might see this technology deployed?
Yeah, you know what? Can you just repeat that last question? My network just popped onto the fast network, so now you're not talking slowly. So if you could just repeat that last question for me, I'm sorry. That's great.
No, that's fine. mRNA was such an amazing technology, and you know, to have been able to deploy it for COVID. Looking into a non-COVID future, do you think there are candidates for other things that we might see this technology deployed?
Yeah, I would be shocked if we don't see it used for everything. It'll be tried for everything from influenza to several other infectious diseases. I'm a little more sanguine on the technology than some might be. I think it's probably going to be the right technology for certain things, just like certain other viral vectors are the right things for others. So, but I think to the extent that we can leverage it, if we can leverage it for influenza, that would be amazing. The reason why is that it's so much easier to manipulate the sequence of an mRNA vaccine when you need to alter it from year to year than it is to deal with what we currently do. For those of you who aren't aware, most of our influenza vaccine is made in embryonated eggs, so chicken eggs, and we have to adapt the virus to grow in those eggs. It's a whole big process, which means if you have a new strain of influenza, it takes months to get there. Whereas because you can actually chemically synthesize this stuff, the RNA, if you wanted, you can change things within weeks.
Great. Well, it explains then when I was working in the vaccine clinic at my organization, whereas I was used to hearing the question about, do you have an egg allergy? Now they were talking about propylene glycol.
Yeah. So right, the polyethylene glycol and several things that are like it this was an allergy issue. That's exactly right.
Yeah. I'm going to turn it over to Joe. I think Joe had a question that you posed there in the chat, and then some other things that we wanted to talk about.
Yeah, I did. I threw something in the chat as a follow-on to one of your responses there, Peter, and that's just to encourage our attendees to go ahead and pop your questions for Dr. Marks into the chat, and we'll be monitoring. We'll make sure that your questions get answered. Just a follow-on in your discussion about emergency use authorization versus BLA, and whether the FDA uses or leverages what we hear is real-world data to supplement controlled clinical study data to make a decision for full licensing.
Yeah, so it will be used for these vaccines. It's going to be used supportively. It's already. I would be shocked if it's not going to be submitted in part in support of that Pfizer wouldn't submit. They look at what's happened in Israel. They vaccinated 5 million people. They have tremendously good data on those 5 million individuals. They were all vaccinated with Pfizer's vaccine, and they have, in real world, 94%, depending on exactly how you cook the numbers, but they actually have 94 to 97% effectiveness, which is pretty impressive, confirming out, in the real world what was done in 45 million, 22 million people received the vaccine, right? So it's very cool to see that. And this is vaccination is a great place where real world evidence can be helpful because the large numbers and the simple endpoints of infected or not infected are very conducive to use for real world evidence. Real world evidence at this point doesn't work as well when we have very nuanced endpoints. Like, it's hard to do a study of a neurologic disease using real world evidence because headaches are hard to characterize. Various degenerative neurologic diseases hard to characterize, whereas infectious diseases, the test is positive or negative, or you've got a cough and a fever, or you don't have a cough and a fever. So it's a great place for real world evidence. We've used it before, actually. We used it for understanding whether high dose influenza vaccine was more effective than standard dose influenza vaccine in the elderly. And actually there, it was really pretty nifty. The real world evidence actually agreed with a randomized clinical trial, but even went further, because in the real world, you actually get endpoints that mean something. Like, you keep people out of the hospital in large numbers, which you can't really know. You don't get those kind of answers when you're doing conventional clinical trials.
Oh, that's great. And before we move on from vaccines, we do have other topics for you. I had a question because of the data that you showed, the demographic data you showed in your slides and the studies that were done. It appears at first glance, that perhaps Black and African Americans are underrepresented in the data set, but you explained that matches the demographics in the US.
Mm-hmm.
But this is a global pandemic. So I'm wondering if you wouldn't mind just zooming out for a moment and just comments on the importance of clinical trial diversity and ensuring that the data used to make approval decisions reflects the populations most affected by a disease or those we wish to serve globally.
Yeah, no, I think this is something that we've probably will come out of this. Even we knew clinical trial diversity was important going into the pandemic, and it's very clearly important coming out of the pandemic. People feel much more comfortable if they know that the vaccine has been tried in individuals like them, or that goes for other drugs as well. And particularly for drugs, it's very important because we know that different populations can have different responses to drugs. For that matter, for vaccines, we know that for previous vaccines, certain populations had different extents of immune response. Now, we happen to be lucky with this particular set of vaccines where these vaccines seem to be so potent and do such a good job at generating an immune response that there has not been differences in different racial or ethnic groups that we've seen that are anything that's really notable. But it is true that it can happen with other products, so we have to always be astutely aware of it.
That's fantastic. Thank you. Okay, if there are no other questions about the vaccine, I'm looking at the chat. I don't see anything in there. Would like to move on to cell and gene therapies, which are enjoying a renaissance, if you will. And we're certainly experiencing an influx of companies focused on these therapeutic modalities. So from your perspective, how has the field matured over the years? What's going to be different this time around? And what challenges is the Center for Biologics experiencing in approval process for medications that are as unique as the patient they're meant to treat?
Yeah. So I think we've come to a really interesting crossroad here where certain things are really taking off and other things are still winding their way through. And they're making progress. So let me start with what's really taken off. Chimeric antigen receptor T cells that are made, that are particularly the ones that are taken from a person and then given back to a person. Those are the autologous CAR T cells. We now have 5 different CAR T cells licensed and more probably on the way in the not too distant future. These have made a big difference in particularly in blood cancers, lymphomas, and multiple myeloma. We now understand that the companies that are making them consistently. There are still some challenges, but it's really come a long way in 5 years. And I think it's even transitioning to the next generation products, which will be these same types of CAR T cells, but not made from the patient who's getting them back, made from a healthy donor. And that will make it much easier because it's much easier to have an off-the-shelf product than one that you have to make locally or at a central facility and then return back every time. For gene therapies, we've seen a lot of progress. We've seen the launch of the first systemically administered gene therapy. That is a product for it's one of the more common rare diseases for a disease called spinal muscular atrophy type 1. It usually takes the lives of children by the time they're about 3 years old. This gene therapy stops that from happening, and they develop normally as if they're, and you don't see any evidence of abnormalities in these kids, and that works pretty well. So it's a great model, and now we see other gene therapies coming up behind it. It hasn't worked quite so well for all of the ones, and there have been some challenges, for instance, in hemophilia, which is a blood disorder in which people don't clot normally, and it's due to proteins. There are two different proteins in general that are the major actors that cause this. Deficiencies in factor 8 and factor 9. And attempts to use gene therapy to replace those, they've been successful, but not quite as wildly successful as we might have liked to see. And so those are working their way through, and we're learning a lot about gene therapy this way. We're learning about what is needed to allow the body to accept these gene therapies, and what may be associated with these gene therapies continuing to express the proteins that they deliver. So a lot is being learned. I suspect that in the next couple of years, we'll learn even more and we'll make even more progress. But it is tremendously exciting because at least we have, in principle, with the gene therapy approaches using adeno-associated viruses, which is a very common gene therapy vector being used now, that those adeno-associated virus gene therapies, they're able to address quite a number of rare genetic disorders potentially, and really make a big difference on people's lives.
Well, that's great. As a recovering molecular virologist myself,
Okay.
I do. I've been in the field, last generation, the Achilles heel was the use of viral vectors that were integrating and these
Right.
off-target effects, right? And
Right.
I think that it's smart for the industry to focus on blood disorders, right? Because you could sort of trade a cure for possibly creating another problem. But I do hope that these delivery vehicles will evolve in a way that they're much more useful in capacity and tropism, et cetera.
Exactly.
Do you feel that they, we reached a level of standardization where CBER can evaluate them properly with regards to safety, efficacy, potency, et cetera? Or there are still challenges in the different companies and what they're bringing?
There's still tremendous. I think the good news is, I think from the clinical evaluation, it's less of an issue. The issue is of manufacturing still remains a very large one in gene therapy. And that's because manufacturing these adeno-associated viruses is not the simplest thing. For those of you who aren't aficionados of biopharmaceutical manufacturing, to make these viruses, you have to use cell lines that grow them. The viruses obviously kill the cells as you're making them. So you have this very awkward situation. Then you have to purify out the viruses from this soup of dead cell debris. And then you have to hope that what has been made properly, and that all the viruses are essentially packaged exactly the right way, and not they don't have too little contents, because that can actually be a problem. And so it turns out that it's been a challenge getting that consistently done. And so we're actually working with NIH, the National Center for Advancing Translational Sciences. There are projects ongoing. We're doing it here in the United States. The Europeans are doing something very similar, trying to help float all boats by having essentially programs that have multiple manufacturers come together to look at better ways to manufacture these products.
That's great. Thank you for that. So I'm looking at the time and I'm looking at the chat window. I'm going to kick it back over to Leslie for our third topic.
Sure. Peter, you know, we're glad that someone eventually persuaded you to leave New York, and that your travels eventually found your way here to Maryland. So you're one of us. You're right here in Montgomery County with FDA over there in what we call the East County, or the Route 29 corridor. And so I would assume that your organization faces some of the same challenges and issues that all of us do in terms of workforce and talent attraction. Just what's happening over at FDA that we should know with you as a major employer.
Yeah, so we are a pretty big employer. I mean, our campus has grown. Before the pandemic, we would have about 10,000 people on campus on a Tuesday or Wednesday, which were our busiest days. A little less on the closer to the weekends, as for many. But we have a very vibrant workforce. It has gone much virtual during the pandemic. Probably on any given day, we only have between 500 and 1,000 people on campus now. We're pretty socially distant. It's mostly people working in our laboratories, mainly because through the magic of conferencing, video conferencing, or the apps like Zoom, et cetera, we're able to get a lot of work done remotely. I mean, our systems are accessible. Most of our documents are electronic anyway. And so people are doing a pretty good job working remotely. It's in some ways it's a little bit of they're good. We take the good with the bad. So what's the good? The good is that we can have more work at home now, I think, coming out of the pandemic. Which will help us because we were running out of space very quickly on in our Montgomery County campus in Silver Spring, where parking was getting to be a problem, et cetera. So this may help us deal with that issue. It may also help us get to retain the best workforce, because sometimes people, as they got towards retirement age, they would retire a little sooner rather than later because they wanted to move away from the area. Now we can actually let people move away from the area to perhaps their retirement area that they want to go to, and still keep them working for a few years. And those are very productive people because they know things. They're very senior and tend to know what's going on. So we have a lot to take advantage of there. It will be interesting. I think the downside to this is that I don't know about your organizations, but there is something. It's been 14, 15 months now, and there is something about not having people in a room together that does create more distance. And it means that as managers, a lot more effort has to go into keeping people I'm trying to find the right word that's politically correct, and keeping people focused, collaborating, cooperative, and avoiding people kind of going off in their own direction. So it will be a challenge, I think, to get the right balance here, because I'm not one of those managers that thinks that come January 1st, it's going to be just like it was beforehand. COVID goes away, everyone will go. Because I think we're irrevocably changed here. There are people who simply aren't going to come in every day like they used to. But it'll be okay, because we need this. We need the space sometimes, and allow us to stay because some of our buildings, they were starting to tell us they didn't even have space in Montgomery County anymore. We're going to have to go further out in Maryland. And that creates its own set of problems.
Yeah, I see a couple of friends in the educational sector here, Margaret Latimer from Montgomery College, and I would think that environment that you're describing for the future is going to be particularly challenging to onboard new employees. How do you create that culture when folks are not necessarily interacting? So what thoughts has FDA been giving in terms of your talent attraction and bringing on your future workforce?
Yeah, so that's really great question. I mean, what we're thinking about is how long, how do we have a nucleus of people, senior people around that will help essentially, um, initiate those who come on board, help them understand our culture, help them to understand what we expect. And probably it will be a matter of people will try to attract people by saying, hey, you need to be on site a certain amount to begin with. And as you get more experience, you can start to work more at a distance. So, I mean, it does take about 6 to 12 months for a new reviewer to start to actually understand how to do a review. And it may be that over that course of time, we'll have people, come into the office most of the time. And then after that, we might be able to let them um, start to work more at home. The good news is that usually in a year's time, in my experience is most of the smart people who work for us, they've started to get the culture by then.
Mhm. That's great. Gigi, we haven't given you a chance to ask some questions.
Well, I love where all the questions have gone. I mean, it doesn't get any better than this, that to have Peter Marks here and to have Leslie representing Johns Hopkins and of course Joe Sanchez with his point of view from AstraZeneca. I mean, how fortunate are we? I'm wondering about the future workforce a different way, which is about training the talent of the future and the degree to which this up and coming field, more data analysis. I mean, how do you get to the solutions? I mean, they've got to leap out of the test tubes, right? Somebody's got to actually crunch all that data uh, somehow, right? And we're hearing a lot about, quantum computing is the way that we're going to be able to crunch all this huge volume of health data, scientific data, and I'm wondering what you're thinking about that. Is that how that's going to impact your planning for the future, how you solve problems, the kind of talent? Are there new career possibilities for getting to FDA?
Well, certainly there is the career pathway of bioinformaticists because we are now increasingly using people who are very skilled in computer programming and in understanding how to take biologic applications and apply them to computers. We are using supercomputing. Excuse me. We're not quite using quantum computing, to my knowledge, at this point, but we are certainly using supercomputing. We do have collaborations with names you would recognize, including its public record, IBM Watson in this area, because there are so many things. When you start to think about getting data on 200 million Americans and starting to crank through it, you need to be thinking about supercomputing. But I'd also say there are other places where there's even more data that comes in, believe it or not. And that is when we're starting to look at these kind of multi-parametric analyses of manufacturing processes, people trying to understand and try to figure out through an incredible swath of data coming in, what is the critical parameter? And that's something that artificial intelligence, machine learning can actually do in a way that we simply can't. Okay, the temperature is X, the pressure is Y, the humidity is Z. We can't, and it changes constantly. We can't put together what actually is most consistent, gets you to the best product. But if you understand what you're measuring in the product, and then all these other parameters, you can potentially start to get better controls on things. And it turns out that kind of data analysis, the amount of data that comes in with that, actually makes the stuff we do for Sentinel monitoring people look like child's play, because it's just so much. It's so data dense, or can be so data dense.
So I was glad to hear artificial intelligence because that's what, I mean, I think, when people thought about artificial intelligence, they thought about the Jetsons and, the robot cleaning up around the house and that kind of thing. And artificial intelligence, isn't that Zumba thing on your floor or whatever that little Roomba is
Well,
called.
many of these things have really enhanced our ability to do our jobs. For instance, we've spent a lot of time with natural language processing because it turns out that that a lot of our adverse event reports, which used to require, I mean, we get thousands and thousands of these, used to require a medical officer looking at them. Natural language processing, now we have algorithms where they can get it something close to 99% right to sort out, okay, a medical officer it flags for the medical officer. And yes, you do probably miss occasional ones, but the idea here is that since you can actually do the work as opposed to having a backlog of months and months, it's worthwhile to go down that route.
Does that mean it'll help with cross-cultural communication? I mean, when scientists from different places are working together, I mean, does it help with that too?
I don't know how to exactly apply. I don't know exactly apply it to that. Although, but I would suspect that we will see a lot of use of natural language processing. We've tried a lot of different experiments trying to understand whether you can even use social media to get adverse events. That didn't go so well, but that's for another story for another day. But I mean, I think there's a lot of places that and computing has become it's become part of a basic tool of what we do, and supercomputing is becoming an increasing part of what we do. We have actually a group in our center called they're called the Hive. It's a high performance virtual environment, and they constantly are pushing the envelope there.
That's exciting stuff. The future is now, it would sound like, at FDA. And we want to just give you another shout out. I mean, you are often referred to as the COVID quarterback for the federal government. Yikes, you cringed. But I know it's met with great affection and respect. We want to also acknowledge again that I know that you were a finalist for one of the Sammy Awards in a whole new category, COVID response.
So thank you. No, thank you. It's been a privilege to try to help out during this. These are things we hope not to live through, but it's what we train to try to help, and it would be nice once it's over and we're back to normal.
And so with that, I would say once all this is over and we are back or forward to a new normal, we would love to have you come back and give us the next chapter and to tell us some of those stories that you just touched on. You just teased us a little bit with it, but we would love to have you back again. And I hope you'd consider our invitation again.
I sure would. It's really a pleasure. Thanks for the great questions, and thanks for the really pleasant evening. Sorry about the little glitches there, but looks like it's clearing up at least.
With that, I would defer to my wonderful chairs. I want to thank Leslie Ford Weber and Joe Sanchez for being incredible moderators. And thank you again so much, Dr. Peter Marks. We're excited about what comes next, and we look forward to seeing you again. Thank you for being with us tonight.
Thank you. Be safe, everybody.
Thank you.
Gigi, can I jump in with one very quick question for Dr. Marks?
Sure.
And I hate to put you on the spot. I put this in a chat, but I know chats can keep scrolling. We are working to get some student days declared at the mass vaccination site at the Germantown campus of Montgomery College. By the way, I'm the vice president provost at Germantown. I loved the comments that you made. One was that we need to stay ahead of this, but I love the analogy that even a runner in first place accelerates as he or she approaches the finish line. May I use that as we roll this out and quote you?
You sure can.
Thank you.
That's not going to get me in any political.
I thought that, but I'm
not going to get me in any political hot water.
I think you're pretty careful about how you get quoted, and I wanted to give you the opportunity to say no. So I appreciate that. It's a great analogy for kids, I think, young students.
My pleasure.
Thanks, Margaret, for asking that question, because now could all of us have permission to use that? Great quote. I think we want to use that. Absolutely. We loved it. My husband's a runner. He was listening to you.
Feel free.
Thank you. Great presentation.
Thanks a lot.
Great.
Thank you.
Thank you, everybody.
You again. And thanks everybody for joining us, and stay tuned. We'll see you again soon. We will be broadcasting or putting this on our YouTube channel. So once it's ready, we'll send out a link. Thank you again, Peter. We really enjoyed it. Thanks so much.
Take care.